Hi, my name is Shane,
I'm Dheeraj.
and today we're going to be talking to you about herpes and its interaction with the human immune system.
So, herpes is a double stranded DNA envelope latent virus, and it has three subfamilies. So, there's the alpha family, which is neurotropic and infects the nervous system. There's the beta system, which is lymphotropic and affects the lymphatic system. And the gamma family, which also affects the lymphatic system and is tumor associated. So there's the side thing with cancers and tumors. And so this is what a typical herpes virion looks like. There's the lipid envelope, the tegument, which is the kind of multicolored part of the virus. And this one has the VP16 protein, which helps form the DNA into circular formation in order for replication. You have the nucleocapsid, which is the black outline of the pink, and that's used to hold and protect the DNA. And then you have the DNA in the center, and the lipid envelope is kind of the peach-colored ring around on the outside. And so the herpes life cycle. So the virus enters the cell by either fusion or receptor-mediated endocytosis, and it more often enters through receptor-mediated endocytosis. And then it uses microtubules to travel to the nucleus. And once in the nucleus, it releases its DNA and uses the VP16 protein, which is in the tegument that we saw earlier, to package the DNA into circular capsids. The virus will also block the production of host cell proteins, recruit transcription factors in the cell for its replication, and it starts the transcription of genes called immediate early genes, which are used for the activation of other viral genes. The virus will also degrade host RNA, and so that it doesn't have any competition for making its own replication. And so there are three different types of proteins that the herpes virus uses. It uses early proteins, immediate early proteins, and late proteins. We already looked at what the immediate early proteins do. So the early proteins and the late proteins, after they help prepare the DNA for replication. So the DNA replicates as a circular piece of DNA, and this is facilitated by the early proteins. The late proteins will then help assemble the virus and push the newly copied DNA into the capsid. The virus leaves the nucleus and goes to the Golgi membrane, or goes to the Golgi, where it envelops itself in a host membrane, where it has the envelope proteins. So now it has two membranes: the membrane it took from the nucleus, which it used to get its envelope proteins, and a second Golgi membrane. And then the virus then buds out the cell, leaving the Golgi membrane behind and keeping its first host membrane, now ready to infect another cell.
So now we're going to talk about barriers. So the stratified squamous epithelium cells are basically just general skin cells. They're going to create a literal wall that are going to be preventing the entry. And this usually does an extremely good job unless there's an abrasion or a wound of some sort, basically a point of entry. And so epithelial cells and macrophages are going to secrete defensins, which are going to help specifically in this particular case. They're going to target the envelope of the virus. And basically what that's going to do is going to create like a structural leak, slowly leaking out the insides of the cells and creating death of that cell. So macrophages and dendritic cells, which are just resident within your skin tissues, both of them are phagocytic. They're just going to be roaming inside the inner layers of the skin, and if they recognize a foreign cell, they will phagocytize that cell and then later present them for the adaptive immunity. And as the last point on the slide says, this is really kind of one of the main points and one of the main procedures that's used to connect the adaptive and the innate immune system to create a more viable response. And so here we're going to go more specifically into the entry of the virus of HSV. And glycoproteins are going to be located, as you can see there on the graph, they're going to be located on the envelope of the herpes virion. And these glycoproteins are going to be GB, GD, and GC. So GB and GC are going to bind to heparin sulfate that's going to be expressed on a wide array of cells, on a wide array of cell surfaces. And this is going to lead to also a wide array or a non-specific binding of host cells. So it's going to give it the ability to really enter a large amount of cells that are kind of located all over the body. More specifically though, or for specific binding, GD is going to bind to cell surface receptors nectin, HVEM or HVEM, and 3-O-sulfotransferase. However, both of these pathways are going to lead to a conformational change in GD. And once GB, GH, and GL, which of all are going to be needed to be activated at the very same time, will, once activated, will lead to fusion or more prevalently receptor-mediated endocytosis, as we talked about before.
So talking about the innate immune system, a recent scientific journal published an article that said that TLR2 may sense the HSV virus at the surface. So this is one of the TLRs that we can use to find this virus. Another one, and more commonly known, is during receptor-mediated endocytosis, TLR9 in the endosome will detect the herpes simplex virus, and it acts through the adapter MyD88, and this leads to NF-kappa B, which, as you know, leads to inflammation, and also type 1 interferons, interferon alpha and interferon beta. It leads to the transcription of these interferon genes. And so once NF-kappa B enters the nucleus, it begins to drive the transcription of for TNF, alpha, IL beta, IL-6, and IL-2, which are all potent cytokines that promote T cell proliferation and differentiation into effector T cells. And so the interferons lead to many effects, but some of the most important effects are the ones that lead to the ISG, or interferon-stimulated gene products, which can be used to block infection, block viral transcription, induce the adaptive immune response, inhibit translation, degrade viral RNA, and for apoptosis. There are also cytoplasmic DNA sensors, which could sense foreign DNA, especially if the virus enters through fusion. And some of these include DAI, AIM2, P2O2, IRRFIP1, and DDX41. And so these all sense double-stranded DNA in the cytosol. So DAI1 produces the type 1 interferon response, AIM2 triggers caspase-1 activation and the inflammasome, which leads to apoptosis, and they also produce IL-1 beta and IL-18. The P2O2 acts as a check factor for AIM2, which means that it prevents AIM2 from activating the caspase and inflammasome if it's unnecessary. So it just kind of keeps it in balance in that way. And then LRRFIP leads to more interferon-stimulated genes, and DDX41 senses pathogenic DNA. So another DNA sensor is called STING, and this activates by double-stranded DNA through the cGAMP, and which will activate IRF3, which will enter the nucleus and induce expression of type 1 interferon genes again. And dendritic cells that have phagocytosed the herpes virus will travel to the lymph nodes, where they will present their antigen to the CD8 alpha positive dendritic cells. And these are the main dendritic cells that will prime the naive cytotoxic killer T cells. And this is the bridging of the gap between the innate and the adaptive immunity. And also, we haven't talked much about complement, but complement activation, for the most part for this virus, is inhibited due to herpes using its glycoprotein GC to bind and inactivate C3b, which will block any further downstream effects of complement.
So I'm sure that you guys remember, but just to reiterate, MHC1 molecules that are going to be on the surface of infected cells, which could be both the macrophages and the dendritic cells that are roaming around in the skin cells that we talked about earlier, they're going to be used for presentation to those cytotoxic CD8 plus T cells. And once those CD8 plus T cells recognize these molecules, they're going to induce apoptosis. And antigen presenting cells, or APCs, also using MHC2 molecules, are going to present antigens via CD4 positive cells, and that's going to start the recruitment of the innate immune system, such as macrophages and neutrophils, but it's also going to help activate the B cells. So there we're seeing another bridge from the active and the adaptive immune system kind of working together in tandem to make a response. Now, after being activated, however, B cells will begin to differentiate into effector cells. Now, these effector cells are called plasma cells. Now, these plasma cells will secrete antibodies in the response to the molecule that's being presented. However, we've noticed in the scientific studies that it really isn't an effective form of prevention, and it doesn't really seem to work. But however, theoretically, it should prevent binding to any of your host cells. And IgG1 and IgG3. These are going to sensitize, basically activating NK cells. So they're basically going to prime NK cells to spit that acid-like quality that we talked about earlier on in the course. And we wanted to mention NK cells in the adaptive immune system, not because they are adaptive, we just want to make that clear that they are an innate immune cell, but we just wanted to again highlight the bridging of both the adaptive and the innate immune system.
So what makes the herpes virus different from other viruses and what makes it so difficult to treat is that it establishes latency. So what HSV does is it enters your peripheral neuronal system through the epithelial cells where it infects on the upper lip, and it will insert its DNA into a plasmid form right next to the nucleus of those neurons, and this is how it establishes latency. And as a result, because the neurons are an immunologically privileged site, it would be more dangerous to have an immune response there than to not have an immune response there, even with infection. So as a result, the HSV remains safe and it can evade the immune system. And then whenever a patient who has HSV is stressed or they're immunocompromised or they have simultaneous infections, this can lead to reactivation of the latent virus, where the virus moves back out of the neurons, back into the epithelial cells where they'll replicate and cause another infection.
So this is just a visual way of expressing what Diraj just told all of you. If you'd like to take a further look at it, just go ahead and pause the video here. It basically just in a more clear-cut way explains the mechanisms that we've been explaining. So memory actually does play quite the role in HSV infection. And basically, so as we talked about earlier, the B cells are going to differentiate into effector plasma cells, but however, they're also going to differentiate into long-lasting memory cells. So what that means is that they're pre-primed cells ready to activate and ready to respond to the virus in the immune system. And basically, central memory B cells are going to help resupply the effector memory B cells, because the B cells are eventually going to undergo apoptosis as their processes run out of energy, start to degrade the cell. And T cells that have also been activated will then migrate to your secondary lymphoid tissues, your bone marrow, and there they're going to just lie in wait for another infection. And memory T cells are able to fight off HSV in the epithelia. However, HSV latency makes it extremely difficult to eliminate this virus, as we kind of mentioned before. And so the picture that we were trying to paint here is that although the virus does establish latency, your immune system does do a very good job at making sure that it has an effective immune response. And basically, the way that you can think about it is that HSV is kind of playing hide and seek with our immune system. So as it presents itself outside of your immunoprivileged sites like your neurons, it is taken care of swiftly, it's taken care of immediately, although you will notice kind of aesthetic things like the fever blisters on your mouth or maybe in your genitalia area. But however, the prognosis for HSV in general seems good. Unless it enters your bloodstream, where we talked about earlier, where complement can be deactivated and cause viremia. Other than that, though, it really is just kind of an aesthetic bummer. Other than that, though, you can live with this virus. You can continue your everyday, daily life with this virus. And thank you for listening to our presentation. My name is Shane.
I'm Diraj.
And this has been the herpes virus.